Hello, Dr. Holden. This is our lesson 2. It's actually going to be the first of 2 lessons on cell organelles. And these ones, the 7 that I have focused in on here, the endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, vacuole chloroplast, and ribosomes, are the ones that the AP College Board said we need to focus a little bit more on. And it is that the students can repeat very nicely what the organelle is, but really do not understand what the function is of the organelle compared to what happens to the cell if something is damaged or something has happened to one of those organelles. In the next lesson, the 3rd lesson, we'll go over the other organelles that you probably would have learned or should have learned in your CP Bio course. So let's start with the ribosomes. So the ribosomes are in every living organism. So they're found in bacteria, prokaryotes, they're found in plants and animals, and they're thought to be one of the earlier evolutionary organelles. And so that will come up in the course that there's going to be questions as to what are the evolutionary factors with organelles? What proof do we have that some evolution has happened? And ribosomes would be one of those. So the ribosomes have ribosomal RNA, and they manufacture protein. And remember that they are both free-floating, and those also manufacture protein as well as being on the rough endoplasmic reticulum. And the rough endoplasmic reticulum does what we call compartmentalizes the cell. And so it has what we call a lumen, or it's a tube. And so when things are manufactured within that, they then would have to leave that tubing. The smooth portion of the endoplasmic reticulum we would have really spent very little time with on your CP Bio class. And so one of the more important functions of that is to detoxify. And it detoxifies by adding one of those hydroxyl units, that OH unit that you should have learned in the first lesson, hydroxyl unit. And when they add that hydroxyl unit to a poison that the body is at least seeing as a poison, it is easier flushed out of the body. So we're going to take just a moment and talk about what does the body consider a poison. Well, virtually everything it doesn't manufacture, it views as not belonging. So even the medicines that a physician will prescribe, the body is going to try to remove those. And so any place that you find filtration in your body, specifically the liver is a very large filter organ, you're going to find more cells with this smooth endoplasmic reticulum in it. And what happens is that the longer the body recognizes that it has something that it has to detoxify, the more of these cells that you're going to have. So taking a look at the liver, alcohol or barbiturates are both considered to be poisons as far as the body is concerned. And so it tries very hard to remove those from the body by flushing them out, by adding those hydroxyl units. So the problem happens with a person that is becoming alcoholic or does some binge drinking in college. One of the reasons why I say be very careful about those fraternities and sororities is because when you have an initial binge of alcohol and your body is not used to that, you're not an alcoholic, you have not built up all these extra cells. Your body actually has no ability or lacks the ability to be able to detoxify quickly. And that's when the alcohol overdose happens and the person can easily overdose and die. But also the regular prescribed medications are treated the same way. So if a person is given a physician gives them a prescription for medicine, if they're on that same medication for a year or 2, what happens is the body has built up a tolerance to it because it has built up more of these cells with that smooth endoplasmic reticulum. And so what happens is then the doctor may have to up the dosage. And of course, if this is going on for years, your body is dealing with more and more of this. It's why many doctors try to hesitate before prescribing somebody a medicine. They'll try to give them other treatments first. Then we have the other function here is it manufactures lipids. So we talked about that as one of the large macromolecules. So that is where lipids are manufactured. And remember, in that lipid category are steroids. And so the steroids that would be manufactured here are your sex steroids, testosterone, estrogen. So you will find more of this also in the ovaries and the testes. A third thing that you would not have known probably at this point, is that that's where calcium is stored. So our body needs calcium at the end of all of its nerve endings, or otherwise the nerve function doesn't happen. So extra calcium is stored there so the body doesn't have to cannibalize its own bone for that. So 3 big functions of our smooth endoplasmic reticulum are detoxification, has lipids and steroids, and stores calcium. The Golgi apparatus is a series of flattened, they look almost like discs. The textbook says pita bread. And they are responsible for, if we have proteins that are manufactured at the ribosomes, they go into that Golgi apparatus, and the Golgi apparatus will make sure that the folding is correct. Remember, we talked about the primary, secondary, tertiary folding of proteins, and that if something happens to the protein, it can become easily denatured and break down. And it can also chemically modify those newly made proteins. And I think the easiest example to explain to students is, here the ribosome is like a cloth factory where they are making bolts of fabric. And if you went and bought that bolt of fabric, that's whoop whoop, but you can't do much with it. So then the fabric is sent to the manufacturing company where it is cut to make tops, or it's cut to make pajamas, or the same bolt of cloth can be making a dress. And so every once in a while I will see someone wearing fabric and I'm recognizing, wow, I've seen that in some other form. And that's because fabric is mass produced and oftentimes sold to a variety of companies. So manufacturing would be here. And if you had taken a course, college course, even a grad course just 8 years ago, they would have spent just a few minutes on Golgi apparatus. And just within the last few years, they've discovered that the Golgi apparatus really has a much greater role as far as proteins than had been realized before this. So now we're going to go on to the mitochondria, which you have a little bit of exposure to in our CP Bio class. And if you had that CP Bio class last year, unfortunately, we usually time the cellular respiration and photosynthesis towards the end of the class. And so, unfortunately, we were on remote learning at that point. And so you did not get the education I would have liked for cellular respiration and photosynthesis. So the cellular respiration, especially with the mitochondria, we'll spend a little bit more time on. Ordinarily, we would have spent an entire 3 days on that in class. So you figure, 80 minutes times 3 would have been quite a bit of time we'd spend on that. But the mitochondria is a double membrane organelle, and the outer membrane is remember that glycolysis would happen outside in the cytosol. And when it enters as pyruvate, it goes through that outer membrane. The piece here that we always see as a fold is the matrix, and that is where your cellular respiration, your Krebs cycle happens. And then in the inner membrane is where the electron transport happens. So again, this is where ATP is manufactured. The lysosomes we would have called, in fact, if you had me, I would have drawn them as little Pac-Men. And the AP College Board has warned that they're reading more and more essays where students put down that the nucleus is the brain center, or that lysosomes act as Pac-Man. And so they're asking us to be much more careful about how we present these organelles, because sometimes students actually write those down in their essays. So we will focus in on a non-Pac-Man type shape, but they have several different functions. So the lysosomes, number 1, contain hydraulic enzymes. 2, they're for digestion and recycling, and 3 for programmed cell death called apoptosis. So let's focus on the first thing, hydraulic enzymes. So if a person is born and they are missing one of their a gene for one of the lysosomes digestive enzymes, and there are at least 50, that it is believed that there's 50 to 55, there might even be more, then the person will have a disorder. For example, Tay-Sachs is a disorder of people that is predominantly of a Jewish background, and it is felt because during periods of persecution in Russia, Germany, Poland, the Jewish people were confined to villages or other locations where the gene pool became a little bit more limited, and somewhere along the line a mutation happened. So if a child is born with missing one of those digestive enzymes, they can develop what's called Tay-Sachs. And in Tay-Sachs, they're missing the enzyme that breaks down lipids, especially in the brain. So the baby looks perfectly normal for their first 5 months or so, and then they are starting to learn to sit up. They are learning how to roll over. And then all of a sudden, things sort of start to degrade, where the baby is no longer developing quite like is expected. And oftentimes medical testing will show that they are missing that digestive enzyme. And unfortunately, there is no real treatment for that right now. And so what happens is that the lipids build up and the baby's brain degrades. And they usually have a life expectancy of about 3 or 4 years. Then again, a second job of the lysosome is that it goes through what's called phagocytosis. So if there is items within the cell that the cell needs to get rid of, the lysosome can actually break those down. And if the item is still large, then that plasma membrane can engulf it and through endocytosis remove that. So again, it helps break down debris within that cell. It can actually recycle some of the material that it breaks down as well. And third, it is what is called programmed cell death. So if the lysosome is given a signal that something has gone wrong with the cell, or the cell should have mistaken it to begin with, it can actually cause the cell to break down, apoptosis, and kill itself. And here we have vacuoles, and I've drawn many vacuoles within the animal cell. If I had a plant cell up here, there'd be just one large central vacuole. But the vacuoles are for storage. So it can be a positive storage of macromolecules that we have excess in the cell and the cell wants to hang on to them. For example, if they want to store in the liver glucose in the form of glycogen, that would be in our vacuoles. They also can be for waste products and other fluids. In the plant, there's only one thing that it stores, and that's water. And that helps, it's one large central vacuole, and that is used just for the water storage and helps establish what is called turgor pressure in the plant, or that nice firmness in the plant. And so the large central vacuole will release water if there's too much, and would try to pull in and retain more water if it is capable of doing that to keep it turgor. And our last one that the AP College Board has asked us to focus in a little bit more is chloroplasts. That would be just in the plant cell or in photosynthetic algae or bacteria. And the chloroplast has a double outer membrane. And again, that was one of the organelles we talked about, but we did not spend as much time on, just like the mitochondria, as we would have if we had not been in remote learning. So that double outer membrane is where our photosystem 2 will occur, or the beginning parts of photosynthesis. And so it is very important as far as without a chloroplast, or if something happens to the cell and the chloroplasts break down, then the cell cannot go through photosynthesis and the cell is going to die. Just like if we are missing a digestive enzyme in the lysosome, there will be some sort of disorder. The person may survive, but there will be lifetime effects to that. And if we talk about ribosomes, there are certain biohazard materials. There are certain terrorist type material that affect the ribosomes. And since all of the protein is manufactured through ribosomes, you can understand quite quickly what would happen if a material was released that broke down the ribosomes so that we can't have protein reduction, protein synthesis. So that would be a definite, the cell is going to die. And same thing with if there's something wrong with the Golgi apparatus, then all of the corrections in the protein folding or changing proteins can't happen. And again, that would have quite a significant effect on the cell. In our next lesson, we're going to go over the organelles that you're more familiar with, and our review of organelles should be finished. Thank you.